Good morning, class 8th. Another part of your lesson 2, that is vegetative propagation. This is also asexual means of reproduction in which a single parent can produce new individuals. Vegetative propagation is seen mostly in flowering plants, angiosperms, especially in those plants where seeds produced are not sufficient or not viable, means capable of forming new plants. So such plants have vegetative means of reproduction where any part of the plant leaving the flower like roots, stems, leaves can produce new plants exactly like the parent plant. So in this means of reproduction we have first vegetative propagation by roots. These are natural ways of reproduction which the plant carries on its own. So in vegetative propagation by roots, certain plants like dahlia, sweet potato, these plants roots are, of course, they store food which we eat like sweet potato. So such plants produce new plants through their roots. Like I have a dahlia root over here. You can see new roots arising up here. One plant which will give, these all separated out can produce new plants. You can see one here, the other here, one here. These all, when planted into soil, can produce new roots and a new plant. Now we have stems. Stems is a vegetative part of the plant, and there are 3-4 ways in which the stem can produce. The first is tuber. All of you have seen a tuber. Tuber is a rich underground stem storing food which we are using as a vegetable. The most common example is potato. Potato is a tuber and it is capable of producing new plants through its eyes. These eyes are adventitious buds which when placed in soil and under proper conditions can produce new plants. After tuber we have bulb, onion. Here I have an onion. The leaves arise from this part, and this is the underground stem rhizome which can produce new plants. This is also a stem which stores food and which we consume, underground stem. Then after bulb we have rhizome. Rhizome, a very common example is ginger. Again, these have adventitious buds. It's a horizontal underground stem, and these adventitious buds grow horizontally and produce new plants. It's again a rich source of food which we consume. Then after rhizome we have corm. Corm is found in ornamental plants like gladiolus, crocuses. Here it's like typically like a bulb only, underground stem, but it has got many scaly leaves. Like you may have had arbi, that's a colcasia, that's a corm. So these corms, when separated out from the parent corm, can produce new plants. Then we have subaerial stem called as runners or offsets. These runners are very common, and that's why if a grass is planted in the lawn, it covers the whole lawn very quickly. I have a grass over here, a runner. You can see runners give roots at every node. The roots are visible up here, and that's how they spread so quickly. This is a runner. a grass. So runners may be seen in mint, pudina, in strawberries, in water chestnut, in aquatic plants also. Okay. Then we have natural vegetative reproduction by leaves. Leaves, example bryophyllum. Here the leaf has notches all around it, and these margins, dissected margins, have adventitious buds. These adventitious buds turn into young plantlets, and if separated from the parent plant, they form new plants. Then we have artificial vegetative propagation, which was started by men to suit his needs. So the first method in it is cutting. In this, simple stems are cut into with a node in it. You can see a node with leaf, and they are planted into soil. They gradually develop roots and shoots and form a new plant. So from the parent plant stems, tiny portions can form new plants. Then there is layering. It's seen in jasmine. Here the parent plant is this one, and a part of the stem of the parent plant has been bent and buried in soil. This buried stem develops roots and shoot system, and it can be cut off from here and transplanted to form a new plant. This is layering. Then we have grafting. This is commonly practiced by orchard growers. Here the desired two varieties of plants are taken. The one which is attached to the ground is called as stalk, and the other piece of stem or root which has been obtained from a desired tree is called as scion. Both the scion and stalk are tied against each other very tightly with a slant cut so that their tissues remain in contact. The joint is made totally free of any fungal or bacterial outgrowth by using certain medicines, and gradually the scion fuses with the stalk and a new shoot arises, forming a new plant. So those orchard growers who want to improve their qualities of mango or lemon, like they want to have a plant in a mango which is disease free and another plant has fruits which are very good in quality, nutritious, sweet, big in size. So from the plant which has better fruits, scion is being cut off, and the one which is disease free, that stem is taken as stalk, and both of them are tied together. So the new plant which we will have will be disease free also, as well as it will have better quality of fruits. So this is grafting. Then the last method of artificial vegetative propagation. This method is totally a scientific method used in laboratory conditions called as tissue culture. In this, any vegetative part of the plant, root, stem, leaf, is removed from the parent plant and it is put in aseptic conditions, totally sterilized conditions in laboratory in a test tube or a beaker, and provided with nutrient medium, growth medium. This tissue is called as callus, C-A-L-L-U-S. Gradually it grows and then it is put into another test tube which has growth regulators or hormones, and the callus differentiates into root, stem, leaves and forms many plants, tiny plantlets, many from a small piece of plant tissue of parent. Many new plantlets are formed, exactly copies of the parent plant. So this is practiced by garden planters like chrysanthemum, marigold, and other ornamental plants are grown by the method of tissue culture when hundreds or thousands of plants are needed from one small piece of tissues. So this is economically very useful method of growing plants, especially in seeds with which are non-viable, like grapes, banana. Tissue culture can be practiced. Now, vegetative propagation as such is a very advantageous method of reproduction. Production or propagation is a most important method of reproduction used by plant growers or farmers. Why? Because here there is a quick multiplication of plants. There is no need to wait for pollination, fertilization, growth of flowers. Quickly from any part of the plant, root, stem or leaf, new plants can be formed. So rapidly we can have lots of plants from simple cuttings or tissue. Then all the plants, the new plants, are exact copies of the old parent plant. They will have no change in any part. They will be exactly like the parent plant, whether in color of flowers or type of fruits or type of leaves. And then they are easy to grow. You don't have to wait for the seed to germinate here. The plant is already there, tiny plant. You simply have to put it into a new growing medium with soil, and it develops and starts fruiting quickly. So they are much easier to grow, and there is no loss of plants as such, because the plant is already developed. And then there is no seed formation, so quickly we can multiply the parent plant. Its copies can be obtained very fast, especially in case of banana, sugarcane, and so on. But there are certain disadvantages also. Since the young plants are exact copies of their parent plant, so if the parent plant has any diseases, that will be passed on also to the daughter plants. So if that disease persists, the whole group of plants can perish. Plus, you cannot get new varieties possible. There are no hybrids possible in this method. Then, so, like in vegetative propagation by leaves, many new plants grow together, and this results in overcrowding and lots of competition between the new plants, so that some of them, if not separated quickly into different areas, may die due to lack of food and water and space. So that's all for today. Thank you.